Chapter 18, the reproductive system, and Chapter 19, pregnancy. Here are the primary objectives for this module, and as you can see there's quite a bit of them, so I'm not going to read them, but feel free to pause the video and read them to yourself. Section 18.1, Survival of the Species Depends on Gamete Formation. This section relates really well to Chapter 20, which is what the extra credit quiz was over, so if you didn't take that quiz for whatever reason, I still highly recommend going over Chapter 20 just because it relates so well to this section and ought to better explain it. So moving on, the main purpose of the reproductive system is first to produce gametes or egg and sperm cells, and then to unite these together to form a new individual. And this is all in the context of sexual reproduction as opposed to asexual reproduction where this doesn't happen. So sexual reproduction involves choosing a mate based on phenotype, which are the genes expressed by an individual, as opposed to the genotype, which is all the genes within an individual's cells, regardless if they're expressed or not. And obviously a mate is chosen based on phenotype because we can't see the genotype. And then these genes are mixed together from the two mating individuals to form a new individual. So essentially the ultimate goal of sexual reproduction is to mix the alleles in the gene pool together in order to create new genetic combinations within the resulting offspring. So if I don't say this later, alleles are the various forms that an individual gene can take, and these always occur in pairs in individuals, such that you might have two different alleles within an individual, and whichever allele ends up being expressed depends on dominance and other things mentioned in Chapter 20, and that's why I recommend going over it. But anyway, these new combinations which are produced are essential to the survival of the species. So sexual reproduction serves as a vehicle to facilitate genetic variation, which is not so with asexual reproduction, which is essentially just the cloning of the original cell or organism. This genetic variation is the basis for the adaptation of organisms to their external environment, and this is because some individuals will always be better suited to the environment that they live in, or we would call this more fit than the other individuals in their environment. And this ensures that the fittest organisms survive and pass on their genes to the next generation, thus increasing the percentage of their alleles in the gene pool, which ought to increase the strength of the organism overall, or its ability to survive, I should say. But anyway, all of this is the basis for some of Charles Darwin's theory of evolution through natural selection. Actually, I shouldn't say all. I shouldn't say some of it is. Essentially, all of this is the basis for this theory. If some of that information on the last slide was a little confusing, we're going to revisit that material in the evolution module, so that should help to iron some of it out. Moving on, the reproductive system forms and unites gametes. The passing on of genes requires the formation of these haploid gametes, and haploid just means that these gamete cells have half of the genetic material found in normal cells. So since normal cells have 46 chromosomes, haploid gametes have 23 individual chromosomes and no pairs. So a gamete is essentially a reproductive cell that will form a new individual when mixed with the opposite type of gamete, or when eggs and sperm combine, I should say. And these haploid gametes are produced via a process called meiosis, which is very similar to mitosis, and we'll cover it in more detail in the next slide. And this is a specialized type of cell division that ensures the equal and orderly division of chromosomes. So in order to form these gametes properly, the normal diploid chromosome number must be cut in half such that it's haploid, and with the resulting gametes having exactly half the usual complement, such that there are only 23 individual chromosomes and absolutely no pairs of chromosomes. So when these two haploids gametes unite to form what is called a zygote, the original diploid number is restored such that we now have 23 pairs or 46 total chromosomes within the new individual. And over the course of this process of meiosis, there is no random splitting of the chromosomal content usually, or at least there's no splitting ideally speaking. So these homologous chromosomes come together and are then separated one to each gamete. So these homologous chromosomes are essentially just chromosomes with the exact same genes on them. There'll probably be variation in the genes, obviously, but they have the exact same genes. So, well, I mean, not exact, but they had the same genes, I'm sorry. The two gametes, the egg and sperm, then come together in a process called fertilization. The resulting diploid zygote then has one set of chromosomes from each parent. Here's a diagram of the process of meiosis, and before you start worrying about this too much, meiosis is essentially the same as mitosis, but with a few differences. So at the beginning of meiosis, we've actually replicated the DNA twice, such that we've formed chromosomes called tetrads, which essentially just means that we have 4 copies of each of these individual chromosomes, and these tetrads are all homologous. So once we've got our 4 sets of chromosomes, we can essentially just do mitosis twice, such that when we end up with haploid gamete cells. So in meiosis 1, we essentially just go through one phase of mitosis, and then in meiosis 2, we go through a second phase of mitosis, such that we have half of the normal amount of chromosomes in each of the 4 resulting cells. And I'm going to tell you right now that in sperm formation or spermatogenesis, actually all of the cells, all 4 of the cells created are viable, which if you compare with oogenesis in females, where we actually only end up with 1 viable egg. And another difference between meiosis and mitosis is that we have an event which is called crossing over, which allows for different alleles to be carried into the resulting zygote. And what I'm just saying here is that the genes on one chromosome can switch to that of another homologous chromosome, so long as it's homologous, such that we don't get the same genes in the chromosome each time we carry out meiosis. I know that's probably about as clear as mud, but it's a little hard for me to explain sometimes. But anyway, don't worry too much about that. Here are the final results of meiosis. So when it occurs in males, 4 viable sperm are produced from 2 divisions, or rather 1 round of meiosis, of a primary spermatocyte cell, which we'll talk about what a spermatocyte is here in just a second, and compare this to meiosis, which occurs in females, where only 1 viable egg is produced from 1 round of meiosis. And this is because this 1 egg is going to need all of the cytoplasm and organelles, or essentially all the cytoplasm and organelles found in that original dividing cell. And the extra genetic material that was created is split out and ejected from the developing egg with little associated cytoplasm. And this ejected genetic material will be found in the polar bodies, which are essentially not viable eggs because they don't really have any cytoplasm or organelles, because they're all taken up by that one viable egg. So after these gametes are formed, the second primary function of the reproductive system is to bring them together to form a new individual. And these gametes require care after they are formed. They need to be united in a protected environment, and the resulting embryo needs to be nourished and protected as it develops. And all of this is a couple of the functions or additional functions of the female reproductive system. So there are several other functions of the reproductive system. One, it triggers puberty, which is the process by which one reaches sexual maturity. After we've reached sexual maturity, the reproductive system needs to maintain the reproductive ability and also stimulate the secondary sex characteristics, which we of course develop during puberty. And then finally, it needs to produce hormones involved in sexual maturation, which leads back to puberty again, and also general homeostasis, which is related to the upkeep of the reproductive system. And both the male and female reproductive systems are composed of 4 components. There are the gonads, which are the organs that produce gametes. So these are the ovaries in females and the testicles or testes in males. They need ducts to transport the gametes and any fertilized egg that is present. They have accessory glands which facilitate gamete formation and survival of those gametes within the reproductive system. And then they need several supporting structures to help deliver the gametes and support them. And we'll talk more about this in more detail in the following sections. Section 18.2, the male reproductive system produces, stores, and delivers sperm. So in order for the male reproductive system to fulfill its functions, it requires gonads to produce the sperm, and these are the testes, like I said earlier, tubes to carry the sperm, 3 types of accessory glands to produce fluid to sustain the sperm, and several supporting structures to help deliver the sperm. So essentially, the male reproductive system is just a single tube. The sperm is generated in the gonads at one end, it matures along that route, and is finally released from the body at the end of that route. And these accessory glands add secretions to nourish and carry the sperm before it is released, and we're going to cover all of this in the following slides. Here is a diagram of the male reproductive system for reference, and we're going to cover most all of these parts in the preceding slides, but anyway, one thing you can see in this slide is the urinary bladder and where it connects with the male reproductive system. So you can just see that the male reproductive system has two purposes essentially. One is release of urine and the other one is release of semen. Sperm is produced in the testes. The testes are paired glandular organs suspended in the scrotal sac or scrotum. The main purpose of this sac is to help regulate the temperature of the sperm, because if these sperm cells get too hot, they'll die. And this temperature regulation is accomplished by the muscles of the scrotal sac, which contract to bring the testicles closer to the body to keep the sperm warm when it's cold, and then these muscles relax when it's warm to get the sperm farther away from the body so these cells don't get too hot. And this contraction of these scrotal sac muscles, this is responsible for what's sometimes called shrinkage. The male reproductive organs actually begin development just 7 weeks after conception, forming from the embryonic mesonephros duct. By 7 months after conception, the testes migrate from the abdominal cavity to the scrotal sac. This process is called the descension of the testes, and in addition to the testes descending, so do the associated vessels, nerves, lymph, and reproductive cords. And this descending of the testes is vital to reproductive health. If only one of these descends, we're going to have, well, the male's going to have reduced fertility, and if neither of them descend, the male could potentially become sterile. And each of these testes, which is spelled wrong there, sorry, carries out spermatogenesis independently within the individual pouches of the scrotal sac. The testes are actually a densely packed mass of seminiferous tubules contained in 200 to 300 lobules within each testis. Okay, testis isn't spelled wrong, that's the singular form of testes, sorry. And an individual lobule holds up to 3 tubules. So that's just to give you some idea of how these things are divided. So here's a diagram of the testes and scrotum. So up in the top left hand corner you can see a transverse section of the penis, and also a lot of the tissues associated with the testes. So there's the spermatic cord, and then the vas deferens, and then the arteries and veins, lymphatic vessels, epididymis, and finally the testis, which is singular of testes, remember. And then over there in the top right is just a microscopic image of the seminiferous tubules, and it also shows the spermatids and the various sperm cells, or the various developmental sperm cells, so the spermatocytes and some of the other cells. And then at the bottom is just a drawing of that, where you can see that spermatogenesis, or the development of sperm cells. You can see how these cells divide and develop into the final sperm product, or the mature sperm cells, which we'll talk about in more detail in the following slides. The cells that produce sperm are found lining the seminiferous tubules, and there are two types of cells within the seminiferous tubules. There are spermatogenic cells, which are the cells that are stimulated to begin dividing into sperm at puberty. First, these cells divide into spermatogonia, then they further divide into primary spermatocytes, and they eventually divide into spermatids. And these spermatids function like separate entities, separate from the rest of the male's body. And these cells actually need protection from the male's own immune system. And this is accomplished by Sertoli cells, which are cells that extend from the basement membrane of the seminiferous tubule all the way to the lumen. These cells surround the developing sperm and are responsible for protecting it against immune attack. And then another type of cell found within the testes are the Leydig cells, which are responsible for producing the hormone testosterone, the primary function of which is to stimulate spermatogonia to divide and develop into mature sperm. This hormone also stimulates bone growth, increases hair production, and stimulates cartilage growth of the larynx, and also increases libido. So it's responsible for a lot of the secondary sex characteristics, which explains why men are usually larger and taller than most women, why men can grow facial hair, and why the male voice is typically deeper than that of the female. The process of sperm formation is called spermatogenesis, and this whole process is the making and maturing of a sperm cell, also called a spermatozoon, and this process takes 65 to 75 days to complete. It begins with cells called spermatogonia, which are essentially stem cells, or another name for these cells would be germ cells. So when the spermatogonia divide, they move forward to the lumen to begin the process of spermatogenesis, and every time one of these move forward, they're replaced by a new spermatogonia, and these cells are still diploid at this point. Once these cells are protected by these Sertoli cells, meiosis can begin to occur. At the end of the first round of meiosis, we have two secondary 23 chromosome spermatocytes, so they're haploid at this point. Each of these secondary spermatocytes divides further to produce two haploid spermatids, yielding a total of 4 viable haploid cells. And compare this to the end result of oogenesis, where only one viable gamete results. The spermatids acquire the correct shape and are eventually ejected from the Sertoli cells. When the sperm are free of the seminiferous tubule, they are called spermatozoa, and that's the plural of spermatozoon. And these are fully formed, yet they're not activated, so they can't swim yet, or at this point. So here's the whole process of spermatogenesis, which actually begins in the brain with the release of these different hormones from the pituitary gland, which are follicle stimulating hormone and luteinizing hormone, and also inhibin, which inhibits the process. But we'll talk more about these hormones later and why they sound like they ought to be only found in females. But anyway, these hormones essentially tell the spermatozoa to develop and then divide. So here you can see the spermatogonia, which is the plural form, have 46 chromosomes. They differentiate and still have 46 chromosomes. They develop into the primary spermatocyte, which still has 46 chromosomes, which then goes into the process of meiosis to produce the secondary spermatocytes, which have 23 paired chromosomes still. And then after the second round of meiosis, we end up, or rather the second part of meiosis, we end up with 23 individual chromosomes within each of these spermatids, and these are released from the Sertoli cells as fully developed spermatozoa, though I wouldn't say they're fully mature because they can't move their tails or anything like that at this point. A normal human male actually produces about 3 million sperm per day from puberty until death, which is actually kind of low compared to some other animals. Sperm exists to reach and penetrate an egg cell or oocyte, and each part of the sperm cell is necessary to reach this end goal. So here are the parts of the sperm. There's the nucleus, which obviously contains the necessary genetic information, 23 individual chromosomes. There's the acrosome, which is kind of a neat part of the sperm. It's essentially the tip of the spear. It's the vesicle on the tip of the sperm head that actually contains the digestive enzyme, which eats through the oocyte membrane so that we can push the nucleus into the oocyte. And then there's the midpiece, which is essentially the sperm's engine, because it contains many mitochondria that produce the ATP needed to reach the egg. Of course, swimming through the female reproductive tract requires a lot of energy. And then there's the tail, or rather the flagellum, I should say here, which is used for propulsion. And also, the sperm doesn't really swim through the female reproductive tract so much as it moves forward by sort of flapping this flagellum. And over here you can see this diagram of the various parts. The acrosome and the nucleus are located in the head of the sperm, whereas the middle piece or mid piece and flagellum make up the tail of the sperm. Sperm and semen are transported and stored in ducts. So once the sperm is produced in the seminiferous tubules, it must be transported from the male to the female reproductive tract. So semen is the fluid containing sperm and other ions, which is formed to help the sperm traverse the ducts of the male reproductive system. The Sertoli cells also create a fluid, which is part of semen, that fills the seminiferous tubule lumen and pushes the developing spermatozoa along. The spermatozoa leave the seminiferous tubules in the lobules of the testes and travel to what is called the rete testes, which is actually the network of tubules found at the junction of the testis and the epididymis before they leave the testes. So the epididymis is a tube which serves as a storage area and final maturation center for the spermatozoa. They lose the last bits of their excess cytoplasm and gain mobility and the ability to fertilize an ovum. So the epididymis is essentially where the sperm cells learn to swim. The spermatozoa can remain quiescent in the epididymis for approximately 1 month, or they can remain viable here for 1 month. A small peristaltic wave is generated in the smooth muscle of the epididymis wall, which helps to push the spermatozoa into the next tube in the system, which is called the vas deferens. The vas deferens transports and stores sperm. It runs approximately 50 centimeters from the epididymis in the scrotal sac and runs through the abdominal wall, looping over the ureter of the urinary system and running behind the urinary bladder. The length of the vas deferens is used to store sperm for as long as several months. If there is no ejaculation or release of sperm from the male reproductive system, the sperm are eventually broken down and reabsorbed by the body. The placement of the vas deferens and its function as a sperm transport vessel make it a primary candidate for sterilization surgery, which is called a vasectomy in this case, and this is when the vas deferens is clipped such that the sperm can't run along the length of the vas deferens. Seminal vesicles help nourish the sperm, and these are located posterior to the base of the urinary bladder. They secrete an alkaline fructose-rich fluid that serves two purposes. One, the high pH of this alkaline fluid helps to neutralize the potentially lethal, extremely acidic environment of the male urethra and the female reproductive tract. The fructose serves as an energy source for the sperm as they become motile. The prostaglandins are also released by the seminal vesicles, and these are hormones which open airways, stimulate the sensation of pain, reduce stomach acid production, cause local irritation, and also stimulate sperm motility. There's also, the seminal vesicles also release a clotting factor responsible for the coagulation of sperm after ejaculation, which causes the semen and the sperm within to stay close together such that they can better survive the female reproductive tract. The seminal vesicles also, they secrete 60% of the total ejaculate volume, so that's how much they contribute to the semen. The ejaculatory duct runs through the prostate gland. So after leaving the seminal vesicles, the sperm travels along the short ejaculatory duct to the prostatic urethra. The union of the terminal end of the vas deferens and the duct from the seminal vesicle marks the beginning of the ejaculatory duct. Semen does not reach this duct except during and only during ejaculation. And ejaculation, if I didn't say this earlier, is actually the process by which sperm and semen are forcefully pushed into the prostatic urethra, causing the prostate gland to also add secretions. And the prostate gland itself is a golf ball sized gland which lies immediately at the base of the bladder. And this gland secretes a fluid which includes citric acid for ATP production, proteolytic enzymes to break up the clot formed by the seminal vesicle secretions, and also acid phosphatase, the function of which the book says is unclear, which I'm not sure is accurate or not. Here's a diagram of the prostatic urethra and penis. So up at the very top of this diagram, you can see the bottom of the urinary bladder, and below that is the prostate gland, and within that there is the opening of the ejaculatory duct. And then we come to the root of the penis, which contains the bulbourethral gland, which rolls right off the tongue. And then there's the bulb of the penis. And then we come to the body of the penis, which contains this erectile tissue, which is full of blood vessels. And during arousal, the blood descends down into these blood vessels such that the penis becomes erect. And then finally, this last tube is the urethra, which has a dual function with the release of semen and urine. And we'll talk more about some of these parts later, or in a second. The urethra runs the length of the penis. So once through the prostatic urethra, the semen travels through the remaining portion of the urethra. So immediately upon leaving the prostate, the urethra dives through the urogenital diaphragm. The urethra then continues the length of the penis through the spongy or penile urethra into the external urethral orifice, or the hole of the penis. The bulbourethral gland lies on either side of the urethra at the bulb, or rather the base of the penis, and these glands secrete an alkaline mucus-filled fluid into the urethra, prior to sperm arrival that protects the sperm from the normally acidic urethra of the male, and also acts as a lubricant to ensure that it smoothly passes through the urethra. The penis is a passageway for both urine and semen, like I said or insinuated earlier. The male urethra functions as a conduit for both urine leaving the bladder and sperm leaving the male reproductive tract. So to ensure that we don't have urine and semen within the urethra at the same time, there's a check valve at the junction of the bladder and the urethra, which ensures that only one fluid is present in the urethra at any given time. The body of the penis contains several cylinders of erectile tissue, and these tissues contain numerous blood sinuses which have a role in erection. So during sexual arousal, the arteries that feed into the erectile tissue dilate or fill with blood. Initially, this is caused by the release of nitric oxide from the nerve endings in the penis, and then the release of cGMP, which is very similar to, or it is similar to AMP, and this allows even more blood to enter the erectile tissue. And this combination results in erection, which is an enlarged and stiffened penis, which is necessary for intercourse. The drug Viagra inhibits enzymes that naturally break down cGMP, and this is how this drug works to prolong erections. Now we're going to talk about how the male orgasm propels sperm. So both the male and female sexual responses share some similarities, such as in both sexes, blood flow to the genitals is altered, gland secretion of certain hormones or substances increases, and the orgasm results in the rhythmic contractions of the pelvic muscles. And there are essentially 4 phases to orgasm. First, there's the arousal phase or excitement phase, which in the male, blood flow to the penis is altered, glands begin to secrete lubricating fluids, the heart rate and blood pressure increase, and it is at this point that the male is highly responsive to sensory stimulation, And then we come to the plateau, which lasts from a few seconds to many minutes, and we'll look at that in the chart here in a second. The third stage is the actual orgasm stage itself, which is actually a series of wave-like muscular contractions along with an intense, pleasurable sensation, and this marks the end of the plateau. In the male, orgasm accompanies the ejaculation. And then finally, there's resolution, which begins with a sense of intense relaxation, heart rate, blood pressure, and blood flow all return to normal, and this resolution time is variable. And then we're going to look at differences in orgasm between males and females in this chart. Here are the 4 phases of human sexual response as they relate to orgasm in both sexes. And as you can tell, both of these processes are quite similar, only with a few differences. One being the male's response is more straightforward. So in the male, we have this excitement or arousal phase where we come up to the plateau phase where the excitement or the arousal remains about the same for a little while until we reach orgasm. And during orgasm is when the sperm is ejaculated or ejected from the male reproductive tract. And as soon as that is over, the level of arousal goes back down, and we're in the resolution phase, which I believe can last as long as 15 minutes or so. And then with the female, we have essentially the same thing, but with this main difference: the female can either quickly reach orgasm, and then orgasm multiple times, or not orgasm at all. So here we have this excitement phase and then the plateau phase. And this is for the yellow I'm talking about. And you can see that here orgasm is reached twice, and then in the red it's reached quickly, and then resolution phase is hit early, and this goes immediately back down. And then in the orange we just see the arousal phase, the plateau, no orgasm phase, and then the resolution phase. Male hormones control the rate of sperm production, and this is primarily mediated by the pituitary gland, which lies deep in the brain and is protected by the sphenoid bone and attached to the brain by the hypothalamus. The anterior pituitary gland secretes luteinizing hormone and follicle-stimulating hormone, which govern both reproductive systems. And the reason they have these names is because they were discovered in the female first, and they're essentially the same. Secretion of these hormones is governed by gonadotrophin-releasing factors, which are produced by the hypothalamus. The names of these 2 hormones reflect their roles in females, like I said. So a follicle-stimulating hormone in females stimulates immature oocyte or egg follicles in the female ovary, whereas luteinizing hormone, or LH, stimulates the production of a yellow body. Lutein roughly translates to yellow. In males, luteinizing hormone stimulates the release of testosterone from the Leydig cells. Testosterone has several functions in the male, which include stimulation of male patterns of development in utero. And what the book's saying here is that it leads to the development of such things as the male external genitalia and the proper development of the testicles, for example. Other functions of testosterone include enlargement of male sex organs during puberty, the development of male secondary sex characteristics, the development of sexual function, and the stimulation of anabolism. The secondary male sex characteristics are those typically associated with puberty, so an increase in size and density of the skeletal muscular system, the appearance of body and facial hair, cartilaginous growth of ears, nose, and larynx, which explains why men typically have larger and wider noses than women, and also larger larynxes, thickening of the skin, and increased oil secretion in the skin, which can of course lead to acne. Some tissues of the male actually convert testosterone to a similar hormone called dihydrotestosterone, or DHT, and this is the hormone typically responsible for male pattern baldness, and this is due to the varying sensitivity of hair follicles to the circulating DHT. Follicle stimulating hormone, or FSH, indirectly stimulates spermatogenesis. This hormone is secreted by the anterior pituitary gland in both sexes, and it's saying that FSH indirectly stimulates spermatogenesis because this hormone and testosterone together cause the Sertoli cells to secrete androgen binding protein. This androgen binding protein then moves to the interstitial spaces of the testes via the bloodstream, and once there it binds the available testosterone and helps to maintain testosterone in high concentrations. Testosterone then stimulates the final production of spermatids. However, when the Sertoli cells are functioning to capacity to protect developing sperm, they secrete a hormone called inhibin, which inhibits FSH production from the anterior pituitary, which causes sperm production to slow. So when the Sertoli cells are full and cannot protect any more developing sperm, we have this negative feedback loop. So if the sperm production slows too much, the process reverses. So what causes testosterone levels to vary? Well, one reason is that testosterone operates under a negative feedback loop. So if blood testosterone levels rise too high, a hormone called gonadotrophin releasing hormone, or GnRH, is not released from the hypothalamus as it would be if testosterone levels were low. So when released, gonadotrophin releasing hormone goes directly to the anterior pituitary and stimulates the release of luteinizing hormone. Luteinizing hormone then increases secretion of testosterone by the Leydig cells. However, if gonadotrophin releasing hormone is blocked, luteinizing hormone is not released and testosterone levels will decline. So over here in this diagram, it's pretty much just a visual representation of what we just talked about. So you can see that the hypothalamus releases gonadotrophin releasing hormone, which stimulates the anterior pituitary gland to release either LH or FSH. So when LH, or luteinizing hormone, is released, it stimulates testosterone secretion by the Leydig cells, whereas when FSH is released, it stimulates spermatogenesis together with testosterone, of course, which causes these Sertoli cells to secrete ABP. And anyway, back to the other side. The released testosterone also helps to stimulate spermatogenesis, like I said, but it also causes male patterns of development, enlargement of male sex organs and sex characteristics, and anabolism, which, if you'll recall, is protein synthesis. And DHT also does a lot of the same things, including causing male pattern baldness. So that's essentially all the slides I have over the male reproductive system, a whole 25. That kind of gives you some idea of how much more complicated the female reproductive system is compared to the male reproductive system. And we're actually just going to cover the female reproductive system for the rest of this lecture. So moving on. Section 18.3, the female reproductive system provides and nourishes eggs. So the main functions of the female reproductive system are that it receives sperm, produces eggs or ova for fertilization, provides an area for the fertilized egg to develop into a fully developed fetus, is involved in giving birth, produces the hormones that cause sexual maturity and the development of female secondary sex characteristics. The organs of the female reproductive system include the ovaries, the uterine tubes, the uterus, and the vagina. Accessory organs include the mammary glands and the external female genitalia. The primary hormones involved in the female reproductive system are follicle-stimulating hormone and luteinizing hormone, which, like I said earlier, are both released from the anterior pituitary gland, and the hormones estrogen and progesterone are released from the ovaries. Here's a cross-section of the female reproductive system, and we're going to cover most of the parts here as we move on through the lecture. But one major takeaway that you can see from this, just this early on, is that the urethra in females does not have two functions. It's just part of the urinary system. And you can compare this to the male, where the urethra is essentially a common tube for both semen and urine. And this is not so in the female because they have these two tubes, each dedicated to that one function. The ovaries are responsible for oogenesis, or egg formation. The ovaries produce both ova, or eggs, and hormones. These hormones include estrogens and progesterones. So like we've covered before, oogenesis produces only one viable ovum per meiotic event. The process of oogenesis actually begins before the female is born, so that at birth the ovaries already contain all of the ova she will ever produce in her life. After this event, the primary oocytes undergo follicular atrasia, or degradation, so that by puberty only approximately 150,000 to 200,000 oocytes remain in each ovary, or over about 300,000 to 400,000 between both ovaries. Each primary oocyte sits in the center of a group of follicular cells, which are then stimulated to develop alongside the oocyte during puberty. A primary follicle has 1 to 7 layers of follicular cells surrounding the oocyte. Hormones, and this is FSH and LH, are released by the anterior pituitary gland, and they affect the follicle cells, stimulating their maturation into a secondary follicle and then a mature Graafian follicle. The Graafian follicle bursts during ovulation, releasing the secondary oocyte into the pelvic cavity, and this ovulated ova is viable for approximately the next 24 hours. Here's a diagram which shows the process of oogenesis, both what occurs in the brain and the ovary. So here at the top left is the pituitary gland in the brain, which releases it releases both follicle stimulating hormone and luteinizing hormone. And here it shows how follicle stimulating hormone is involved in the development and maturation of this ova. And as you can see, it stimulates the maturation of the primary oocyte into a secondary oocyte, which still has 23 paired chromosomes. And you can compare this to what luteinizing hormone does, which is leads to ovulation or release of the ova from the ovary. And as you can see, once the secondary oocyte is released, it still has 23 paired chromosomes. However, it develops further and discharges all the rest of this genetic material in the polar bodies, so that we have an ovum with only 23 paired chromosomes, which can then be fertilized by a sperm cell and develop into a zygote. This diagram shows essentially the same thing that the last diagram did, with more of a focus on the development of the follicle in the ovary. So here you can see the primary follicle developing into the secondary follicle by way of FSH, and then you can see the ovum, or the ova, being ovulated. And then here there's a little bit more of a description as to what happens to this follicle after the ova has been released, and it develops into the corpus luteum, which is a structure primarily responsible for releasing progesterone, or the hormone of pregnancy. And this structure is temporary and degrades after some time, and we'll talk more about this in the preceding slides. After the ova leaves the ovary, it enters the uterine tubes, sometimes called the fallopian tubes. The open ends of these tubes are expanded into a funnel shape, infundibulum, which ends in the finger-like fimbriae. These fimbriae collect the ovulated oocyte and sweep it into the infundibulum, and this ensures that it's going in the right direction. These fimbriae do not work like the claw at the arcade, obviously. They sway rhythmically in response to the hormonal controls. The ends fill with blood, descend, and sway, creating small currents in the abdominopelvic fluid, which draws the oocyte into the uterine tubes. The oocyte is then collected in the uterine tube, and the ciliated epithelia lining the tube help wash the oocyte into the uterus. Fertilization must occur within 24 hours of ovulation. The oocyte can travel only the upper 1/3 of the uterine tubes in 24 hours, so fertilization typically occurs here. The oocyte takes 6 to 7 days to reach the uterus and then degenerate unless it happens to be fertilized. If it's fertilized, it will attach to the lining of the uterus, and we'll talk about that more in the preceding slides. The uterus is the womb and is the site of fetal development. The uterus has 3 layers. There's the perimetrium, which is the outer covering, the myometrium, which is the middle layer of smooth muscle within the uterus, and then there's the endometrium, which is the inner lining. This endometrial lining thickens and sheds every 28 days in response to hormone levels, and this results in menstruation. So this is essentially why menstruation occurs, because the woman is discharging the endometrial lining. The implantation of the embryo occurs within, well, it occurs in the endometrial lining. The lining is shed when no successful fertilization has occurred. The cells that line the cervix produce a mucus which helps to aid fertilization. This is a thin, watery, and alkaline mucus during ovulation, which helps the sperm to survive within the uterus. However, this mucus is thick and impassable when no egg is present. And here are a couple disorders of the endometrium. There's endometriosis, which is a disorder where the endometrial lining also appears in the uterine tubes, on the external surface of the uterus, and on the external surfaces of the urinary bladder and other pelvic organs. When this endometrial lining is shed, it is trapped in the abdominal pelvic cavity, as there's no way for it to exit from these areas, and this can lead to lots of pain primarily, and will have to be treated by, well, it'll have to be medically treated. And then there's ectopic pregnancies, which are pregnancies that occur whenever implantation occurs outside the uterus. In all cases, the embryo will not survive, and this tendency to have ectopic pregnancies will lead to, essentially lead to sterility, as will endometriosis. And in ectopic pregnancies, if the implantation occurs in the uterine tubes, medical attention will be needed, as it will threaten the life of the mother. Here's a diagram which shows the anatomy of the uterus, and it clearly labels the uterine cavity and the 3 layers that we just talked about, and also the cervix of the uterus and the vagina. Up towards the right hand corner, you can clearly see the fimbriae of the uterine tube, and then over to the left it points to where the infundibulum of the uterine tube is, and you can see where all this is located in relation to the ovary. And I should say, if I didn't say it earlier, the ovary is connected to the it's connected to the fundus of the uterus by way of this ovarian ligament, and not by the fallopian tube. Some women past reproductive age may develop uterine problems. If these problems are severe enough, a hysterectomy may be required, and this is a surgery which involves the removal of the uterus, and it's typically performed when uterine or ovarian cancer is detected or uterine hemorrhaging occurs. An elective hysterectomy can be used to alleviate difficult menstrual cycles or to remove fibroids, which are benign tumors of the uterus. Severe cramping, bleeding, and other menstrual discomfort are eliminated with the removal of the uterus, because there isn't a uterus there to cramp or bleed or be discomforted. If the ovaries are removed, hormone replacement therapy is usually recommended, and this is because it helps to prevent the postmenopausal symptoms such as night sweats, mood swings, and loss of bone density. However, if the ovaries are not removed, hormone replacement therapy is not required. The vagina, vulva, and many glands complete the female reproductive system. The vagina connects the uterus with the external environment. It serves as the receptacle for the penis, an outlet for menstruation, and also as the birth canal. It is a 10 centimeter long muscular tube lined with a mucous membrane. Walls of the vagina feature transverse folds. The cells within the vagina have a large store of glycogen, which breaks down to produce acids that retard microbial growth. These acids are inhospitable to sperm as well and will kill the sperm cells unless they're buffered by the seminal fluid. Changes in cervical mucus during ovulation and the seminal vesicle fluids help sperm survive the harsh environment, like I just said. The female external genitalia are collectively called the vulva. The most sensitive area of the female external genitalia is the clitoris. This is a small tuft of erectile tissue which is homologous to the glans or end of the penis in males. This is an extremely sensitive area which plays a role in sexual stimulation. So below is just a diagram of the female external genitalia with the, it labels the labia majora, the labia minora, the hymen, the clitoris, the external urethral orifice, and the vaginal orifice. The mammary glands technically aren't part of the female reproductive system, but they're included here just because they serve a major purpose in getting nutrients to the baby after it's born. And they're actually modified sweat glands located above the pectoralis major muscles. They are supported by ligaments and are protected by a layer of adipose tissue. They're composed of lactiferous ducts connected to lactiferous sinuses. Milk is produced in the lobules of the gland, stored in the lactiferous sinuses, and passed out of the breast via the lactiferous ducts. Milk is produced in response to the hormone prolactin, or PRL, made by the anterior pituitary gland, and milk is released from the mammary gland in response to oxytocin. Sorry, I didn't fully type out that sentence. And here in this diagram to the right, you can clearly, it's a cross-section of the mammary gland, which has all the parts labeled. So you can see the suspensory ligament, the pectoralis major in relation to the rest of it, the lobule, the mammary duct, the lactiferous duct, the areola, the nipple, and the adipose tissue in superficial fascia. There are two hormonal cycles which occur in females, and these two cycles occur in non-pregnant women specifically, and they are the ovarian cycle and the uterine cycle. The ovarian cycle is the programmed series of events which occurs in the ovary as the eggs mature and are ovulated, and this cycle is caused by the interplay between the hormones released by the anterior pituitary gland and the hormones released by the ovaries themselves. And then there's the uterine cycle, which is the cycle responsible for the appearance of the menstrual flow, as the endometrium layer is shed approximately every month. And this cycle is controlled by only the ovarian hormones. And a good way to remember the difference between these two cycles is that the word ovarian always refers to eggs, and this cycle is the cycle which controls it's the cycle referring to the development and maturation of the eggs, whereas the uterine cycle always refers to what's going on in the uterus and what growth stage the endometrium layer is in. The whole female reproductive cycle is ultimately regulated by a hormone called gonadotrophin releasing hormone, which is released from the hypothalamus, and this hormone leads to the production of follicle stimulating hormone and luteinizing hormone in the anterior pituitary gland. And we've already went over these hormones in males, so now we're going to go over what they do in females. So follicle stimulating hormone, or FSH, stimulates follicle cell growth in the ovaries and causes the follicles and the associated ova to mature within those ovaries. And then luteinizing hormone, or LH, causes the most mature follicle to burst, which leaves a yellow body of spent follicular cells, or corpus luteum. Remember, luteum is referring to the fact that it's it's yellow, And all of this, this follicle is developing in the ovary. And then another hormone that we've mentioned but haven't went over is estrogen. And this is a hormone typically associated with females. And this stimulates the development of the female secondary sex characteristics, including adipose deposition in the breasts, hips, and abdomen, which explains why males and females look morphologically different. And also, this hormone leads to the development of groin and axillary hair. It also increases protein buildup and increases body mass, which explains why growth occurs during puberty. And it also lowers blood cholesterol. Continuing from the last slide with the hormones typically associated with women, men are capable of secreting these hormones, specifically estrogen. They just don't secrete them in quantities as high as those seen in women. Men do secrete progesterone on occasion. If this hormone is secreted by the prostate, it typically means that prostate cancer is occurring and is causing the hormones to be all out of whack. But anyway, progesterone is usually referred to, or can be referred to, as the pregnancy hormone, and it typically stimulates the growth of and granular secretion in the endometrium. So here it's essentially controlling the regrowth of the endometrium after it's been shed approximately each month, and this sort of relates to its function as the pregnancy hormone, because if this endometrium is not, or rather if the upkeep isn't as it needs to be, then pregnancy cannot occur, because this is where the embryo attaches. And then the final hormone in this section is relaxin, which is the hormone capable of quieting smooth muscle, or rather inhibiting smooth muscle contraction. And this hormone aids in the implantation of the embryo in the endometrium, presumably because if that smooth muscle is contracted, the embryo is going to have a hard time attaching. The production of relaxin increases dramatically if implantation occurs, and this hormone is typically secreted by the placenta. Here's a diagram of the two female reproductive cycles, and I think I actually cut the label off of the one at the top. But anyway, that's supposed to be the ovarian cycle. The one below it is the uterine or menstrual cycle, and the one below that is just a graph showing the hormone concentrations at each stage of both of these cycles, because they're happening simultaneously. So here in the ovarian cycle, we have the follicular phase, which we'll go over in the next slide, which actually involves the inhibition of the follicle stimulating hormone because that's a negative feedback loop. So essentially, the more FSH is secreted, the less needs to be secreted because it's a negative feedback loop. But anyway, that's the hormone that causes the ova to mature and then eventually be ovulated. And anyway, after the ovulation event occurs, and remember that's controlled by luteinizing hormone mostly, or LH. But anyway, after the ovulation event occurs, we have the formation of this corpus luteum, which once again is yellow, and that's why it's called a luteum. And this is a temporary gland, essentially, which secretes progesterone and also estrogens, and then eventually degenerates after a few days, it looks like. And then below that is the uterine cycle, which once again is mostly controlled by progesterone. And you can see that at the beginning, the endometrium has been shed, but will build back up after it's been shed. And this cycle occurs this complete cycle occurs approximately every 30 days. And then below that, and hopefully I can describe this as best I can, is showing the hormone levels at various stages. So FSH, you can see, rises to cause, it rises a little bit at the start to cause that follicle, or rather the egg, to mature, and then drops off, then peaks back up a little just before ovulation, and then goes back down again. And this is essentially, it's more or less the same for luteinizing hormone, where we have this big spike just before ovulation. And remember, that's because this hormone is what causes ovulation in the first place, and then a drop off once ovulation has occurred. And then with estrogen, we have relatively low levels, and then it peaks just before ovulation because, once again, remember, it causes the, it also helps the egg to develop. Then it drops back off and then starts to rise a little bit more to cause the endometrium to build back up. And then finally, with progesterone, it doesn't really do anything until after the ovulation event has occurred, and then spikes back up to help, or rather to cause the endometrium to regenerate. Physiological and hormonal changes which occur during the female reproductive cycles are part of an integrated system. So first is the uterine cycle, which corresponds to that middle portion of the diagram, and this is simply just the regular growth and loss of the endometrial lining, which then regrows. And you can kind of put this off to the side because all the rest of these phases are actually part of the ovarian cycle. So there's the pre-ovulatory phase, and in this phase, FSH secretion increases, and the dominant follicle develops and secretes estrogen and inhibin. And then there's the follicular phase, where this dominant follicle enlarges, thus increasing estrogen production under the influence of luteinizing hormone. And then there's the proliferative phase, where the damaged blood vessels and endometrial lining are repaired and the glands develop in the endometrium. So this, I'm sorry for what I said a second ago, this is actually part of the uterine cycle also. And then there's the ovulation event itself, and this causes, during this phase, we have the increased production of gonadotrophin releasing hormone and luteinizing hormone, and also Also in this phase, the Graafian follicle bursts, which is the ovulation event itself. There's the post-ovulatory phase, where the follicle cells are dormant and the newly formed corpus luteum cells begin to function. Eventually, the corpus luteum degenerates and the progesterone levels drop off, and this is what occurs if no fertilization occurs. And if fertilization did occur, this corpus luteum would remain and continue to release progesterone. And then finally, there's the secondary phase, where the endometrial glands begin to function and the lining is prepared or rather repaired for another possible implantation. So I digress again. The uterine cycle consists of these two phases, the proliferative phase and the secretory phase, and the rest of the phases on this slide are part of the ovarian cycle. Lifestyle has an effect on the female reproductive cycle, and that's not to say that lifestyle doesn't have an effect on the male reproductive system either, but just keep this in mind. The male reproductive system only has essentially two functions: to produce sperm and then release sperm, whereas the female reproductive system has at least 4 or nearly 4 times the amount of functions as the male reproductive system, because not only does it need to ensure that that egg matures and is released and that the uterine walls are repaired, it also provides a housing area for the developing embryo, so that's kind of the reason why the female reproductive system might be more at risk than the male reproductive system. But anyway, this specific condition that we're going to talk about is the athlete or athletic triad, which is a condition in which the health deteriorates, or rather the woman's or girl's health deteriorates due to an overemphasis on sports or other physical activities. And this is actually consisting of 3 different problems. Disordered eating, most likely related to the fact that she would be regulating her weight to compete or participate in these activities. And then there's amenorrhea, which is the actual lack of the menstrual cycle, possibly due to this disordered eating or just over exertion and exercise, essentially just depleting the resources needed to have a menstrual cycle. And then there's osteoporosis, which is the breakdown of the bones just because the body needs the calcium within those bones so badly. The female reproductive cycle eventually stops. So we can contrast the female reproductive system and the male reproductive system in that the male reproductive system can potentially never lose its function. And I say potentially because a man could potentially lose the ability to reproduce due to age, but ideally speaking, this shouldn't occur. But anyway, this point at a woman's life in a woman's life when menstruation stops is menopause. And this actually begins 1 year after the last menstrual flow, and I presume that this year period would be pre-menopause. And this is due to the fact that fewer ova are maturing and less estrogen is being produced. The surge in progesterone after ovulation decreases, and this is responsible for the perimenopausal symptoms, which include irregular periods, hot flashes, mood swings, increased abdominal fat deposition, reduced bone calcium, and loss of breast fullness. And this may last from a few months to a few years. Hormone replacement therapy increases the estrogen levels of the body and reduces hot flashes, which I've been told can be severe. Other drugs may be needed to reduce loss of bone density, and this perimenopausal, one of the symptoms of reduced bone calcium is why older women have the potential to develop osteoporosis more so than older men. Section 18.4, there are many birth control choices. So here we're going to take a break from talking about the physiology of the male and female reproductive systems and talk about contraception. I know in approximately slide 28, I said we were essentially done with the male reproductive system, but we're going to revisit it in these last couple of sections real quick. Don't worry, there's a whole complete chapter on the rest of the issues and topics related to the female reproductive system. So there's still a lot of that. The only absolute method of birth control is abstinence, which is to abstain from intercourse or sex. Other birth control methods rely on either surgery, hormones, IUDs, and we'll cover that acronym in a second, spermicides, barriers, and also some behavior modifications. Each of these forms of birth control has specific advantages and disadvantages. Here's a chart which shows the failure rates of certain types of birth control methods, and we'll just go over a few of these. So the first type of method is no method, which has an 85% failure rate approximately, whereas complete abstinence has a 0% failure rate, which I would expect, no intercourse, no pregnancy. And then there's surgical sterilization, such as a vasectomy, which has a pretty low failure rate, 0.1%. Compare this to a tubule ligation, which is referring to a woman having her tubes tied, which has a failure rate of 0.5%. Another type of surgical sterilization, essentially a hysterectomy, has probably a failure rate of approximately 0%, no uterus, no pregnancy. And then there's some hormonal methods, which include oral contraceptions, intrauterine devices, which we'll talk about in a second. Spermicides have a failure rate that's pretty high. Perfect use is 6%. Typical use is 26%. Barrier methods such as male and female condoms and diaphragms have fairly high failure rates in the typical use of 14%, 21%, and 20%. Whereas periodic abstinence has a similar failure rate to condoms, whether they're talking about the rhythm or the symptothermal method. Now we're going to get into the specifics of contraception, starting with surgical contraception. There are essentially two types of surgical contraception: the vasectomy for men and the tubal ligation for women. We're going to start with the vasectomy. Vasectomies are performed to prevent sperm from leaving the epididymis. This is typically an easy outpatient surgery, in which the vas deferens is first isolated, pulled out of the scrotum, and then closed either by looping or tying or by ligating. And ligating is to either cut it, seal it, clamp it, or otherwise sever the vas deferens. And after they've done this, they'll, of course, put the vas deferens back into the scrotum and seal up the wound. If you remember from the chart, this does have somewhat of a failure rate, and this is due to the fact that sperm may remain in the vas deferens above the vasectomy, causing sterility to be delayed for up to 6 weeks. The form of surgical contraception for women is called tubal ligation, and the goal of this surgery is to block the uterine tubes. And as compared to vasectomies, this is not really an easy surgery, because first the woman must be anesthetized, and then the abdomen is descended with CO2 gas to separate the organs so that they can get to the uterine tubes, and the uterine tubes themselves are then located via laparoscopy, and this will end up requiring a hospital stay for recovery. Once the tubes have been located, they can then be cut, sealed via electrocautery, and this is to basically burn the ends with electricity such that they close. Or the uterine tubes can also be closed with titanium clamps, and also micro inserts may also be used instead. And this really explains why it's much easier for a man to get a vasectomy as compared to a woman having a tubal ligation, simply because it's much easier to recover from a vasectomy than a tubal ligation, as you can see. Hormonal methods of birth control are another option. The birth control pill is an oral contraceptive, obviously, and is actually a combination of synthetic estrogens and progestins. When we say progestin, we actually mean different kinds of progesterone, and these hormones alter the natural hormonal rhythms of the female. There's also the patch form of hormonal birth control, in which the hormones are absorbed through the skin. These inhibit the secretion of FSH and LH, thereby preventing ovulation, or they may alter the mucus production of the cervix to create an environment which is inhospitable to sperm. There is also a form of birth control pill that is capable of providing 3 months of continuous hormonal control. There are also alternative forms of hormonal birth control, such as Norplant, which is a series of 6 hormone sticks surgically implanted, which leaks progestins. There's Depo-Provera, which is an intramuscular injection, which is a shot into the muscle tissue, consisting of progestin, which would be administered every 3 months. And then finally, there are vaginal rings, which are worn in the vagina for 3 weeks and removed every 4th week. And these leak estrogen and progestins. And obviously, with all of these would require a clinical visit. Hormones can also be used to prevent implantation of the fetus within the embryo into the endometrium. There's the morning after pill, which prevents this implantation, or causes the implanted embryo to be lost as the endometrial lining weakens. And this can be administered within 3 days to 7 weeks after unprotected intercourse. There are several other forms of contraception. There's the intrauterine device, or UDI. These are foreign objects which float in the uterus and periodically hit or scrape the endometrial lining, preventing implantation of any embryos which may be present in the uterus. These are surgically implanted and may be replaced every 10 years or every 5 years for those IUDs which leak hormones. Apparently, a few of these have been somewhat dangerous to the individual female's health. If you'll recall from the article in your chapter that you may or may not have read, but apparently most of these IUDs are still fairly safe as far as that goes. There are spermicides, which are creams and jellies, which typically contain non-oxidol 9, which is a compound capable of killing sperm by disrupting their cellular membranes. However, spermicides can lead to the loss of the protective epithelium in the vagina, which can increase one's risk of STDs, or rather the female's risk of STDs. There are several different barrier methods. There is the condom, or more accurately, the male condom, which is a barrier worn on the penis, and this is the type of barrier method with the smallest chance of failure. There are other barrier methods. There's the female condom, the cervical cap, and the diaphragm, which are barriers worn in the vaginal area, and these typically aren't as successful at preventing pregnancy than the male condom is. And then finally, there's the rhythm method, and in this method, one must know the exact timing of ovulation and therefore can avoid pregnancy by preventing the introduction of sperm into the female reproductive tract during that time. And this method actually has a fairly high failure rate compared to some of the others. If those contraception methods fail, the pregnancy can still be terminated through abortion, which can take several forms. I feel as though your book was a little bit light on how serious abortion is, or rather the topic of abortion is, because no matter what your political views on abortion, it's still a pretty serious topic. And personally, and I'm not going to really talk about my own opinion on it too much, because it's such a difficult topic, I have difficulty even figuring out what my political views are on abortion, just because it's such a controversial thing. Abortion actually is simply the termination of pregnancy, either naturally or through some type of medical intervention. So abortions do occur naturally, possibly due to some physical defect in the fetus or some type of fatal mutation, or potentially through the mother ingesting a substance which is detrimental to the fetus's health, resulting in its death. Abortion can take several forms. Early stage pregnancy can be easily terminated using a drug called mifepristone. In this country, elective abortions are performed only in the first trimester of pregnancy and can take one of several forms. The uterus can be scraped clean, resulting in the removal of the endometrial lining and the implanted embryo. The contents of the uterus can be suctioned out. A saline solution can be injected into the womb, resulting in the loss of the endometrial lining. Some abortions are performed for certain reasons. such as a pregnancy resulting from rape or incest, a pregnancy that endangers the life of the mother, or a life-threatening malformation of the fetus. Since this procedure removes a potentially viable fetus, it's very controversial, obviously. However, most states in the United States still allow elective abortions. Section 18.5, Sexual contact carries a danger, sexually transmitted diseases. So sexual reproduction carries a real danger to individuals, sexually transmitted infections or diseases, so either STIs or STDs. STI is actually the more accurate term for these diseases, but STD is still a term used within the medical community. STDs or STIs can be transmitted through intimate contact between the delicate tissues of the reproductive system, and this allows pathogens to move directly from the blood or bodily fluids of one individual to another. These diseases can range in severity from mild discomfort that can be cured with antibiotics to severe reoccurring infections to deadly diseases such as AIDS or Ebola. There are several ways to help prevent STDs. Abstinence would be a failsafe to preventing STDs. Remaining in a mutually monogamous relationship with a person free of STDs is another good way to remain STD free. Using certain barrier techniques such as condoms can help decrease the likelihood of being infected with one of these diseases, though they're definitely not a failsafe. Abstaining from intercourse with people who have tested positive for STDs, or there are also vaccines for certain STDs. However, they're only available against specific ones. Another way to help prevent being infected with one of these diseases would be to not engage in risky sexual behaviors also. This is a chart which shows the estimated new cases of STDs per year in the United States. I'm not going to really go over this much because this is data from 2008, so it's 8 years old. But you can look at this as a reference, and if you want to, you can pause the video to look at it for a longer period. STDs can have many causes. There are several bacterial STDs, which include gonorrhea, syphilis, and chlamydia. However, as these are bacterial, they can be treated with antibiotics. There are also several different viruses transmitted through sexual intercourse, and these include HIV, the herpes simplex virus, or HSV1, and this causes cold sores and actually typically isn't an STD, though it can be. There's herpes simplex virus 2, which is the virus we usually mean when we're talking about herpes. There's the human papillomavirus, or HPV. This virus causes genital warts and may lead to cervical cancer. And then there's hepatitis B, which is inflammation of the liver. Yeast infections can be caused by a fungus, and this is not always an STD. And in fact, if one has a compromised immune system, yeast infections can occur in areas other than the genitals. There are pubic lice, which are insects which burrow into the skin. And then finally, we have vaginitis, which is inflammation of the vagina, which is typically caused by a parasitic protozoan. Here's a chart which shows 6 of the most common sexually transmitted diseases. So feel free to pause the video and read this if you want to. However, I'm going to highlight trichomoniasis, which is the protozoan that typically leads to vaginitis, and it's listed right there at the bottom. Moving on to Chapter 19, Section 19.1. Days 1 through 14 include fertilization and implantation. This chapter concerns itself with all the events related to pregnancy, so everything from the fertilization of the egg to a little bit after the birth of the infant. Prenatal or before birth development has 3 distinct phases. There's the early embryonic phase. In this phase, the ovum is first fertilized by the sperm, and the resulting zygote or early embryo undergoes several cell divisions to form what is called a blastocyte that is implanted into the endometrial lining of the uterus. This phase lasts 2 weeks and ends with the formation of an embryo. There is the embryonic phase, which includes weeks 3 through 8. In this phase, the embryo develops and undergoes even more kinds of cell division. The final phase is called the fetal phase, and this includes weeks 9 through or up until the birth of the infant. The fetus undergoes yet more spectacular transformations and fully develops. Proper human development requires strict and precise controls throughout all of these phases. Fertilization creates an entire diploid genome. Essentially, this is because as the sperm is haploid and the egg is haploid, when the two come together, they create an entire diploid or complete genome within the newly formed zygote. In order for the sperm to fertilize the egg, it must first go through a process called capacitation, which includes all the changes that make the sperm capable of fertilizing the ova. So, in this process, the flagella begins to move faster. The membrane of the sperm head changes so that it can fuse with the ovum, and the acrosomal enzymes within the head are primed to digest the protective layers surrounding the egg, allowing the male DNA to enter the egg. Capacitation typically takes around 7 hours, and this process is helped by the secretions in the female reproductive tract, which facilitate fertilization by degrading the sperm's outer surface. The ova itself is surrounded by several layers, such as the corona radiata and the zona pellucida. The corona radiata is composed of cells from the ovarian follicle. The word corona radiata, I believe, roughly translates to radiant crown, which is kind of a strange name to give it. There's the zona pellucida, which is a clear-looking layer between the corona radiata and the ovum membrane. This layer has specific species receptors for the sperm, and this is the reason why only human sperm can fertilize a human ovum, and explains why hybrids between two species obviously aren't readily created. The sperm binds to this receptor binds to this receptor in the zona pellucida, I should say, and the acrosome activates, releasing its load of digestive enzymes. These enzymes then eat away at the zona pellucida in front of the sperm, while the flagellum continues pushing the sperm forward so that we can ensure that DNA from the sperm gets into the ovum. Only one sperm can make its way into the oocyte. This is called syngamy, which specifically is that only one sperm can fuse with the oocyte. Within seconds after syngamy, the oocyte membrane depolarizes, which blocks the entry of all other sperm, and this prevents polyspermy, or fertilization by more than one sperm. The oocyte finally fully completes meiosis, creating a mature ovum and a small polar body of excess DNA that will degenerate outside the ovum. The male DNA converges into a male pronucleus upon entering the ovum, and the mature ovum DNA simultaneously forms a female pronucleus. The two pronuclei will fuse and create a diploid chromosome complement. Once this occurs, fertilization is officially complete. The entire process of fertilization usually happens in the upper 1/3 of the uterine tubes. The ovum is viable typically for 24 to 48 hours after ovulation. It reaches the halfway point of the uterine tubes in 48 hours. Sperm can survive in the female tract for 5 days and may already be present in the upper reaches of the uterine tubes. All of this just simply is the reason why fertilization typically occurs in the upper uterine tubes. These pictures from the book show where fertilization occurs within the uterus, and also shows all the layers of the ovum that the sperm must penetrate in order to fertilize the egg. And we also have some pictures of the sperm fertilizing the egg. So just as review, the sperm has to penetrate the corona radiata, then the zona pellucida, the plasma membrane of the secondary oocyte, and enters the cytoplasm of the secondary oocyte. The growing human undergoes 3 kinds of cell development. The fertilized egg, now called a zygote, undergoes 3 basic kinds of cell division, and these are cleavage, which is the series of rapid cell divisions which occur in the first 4 days after fertilization. This process does not increase the size of the zygote, but rather divides the zygote into many cells to form a ball of cells roughly the same size as the original undivided zygote. There's differentiation, in which the cells begin to specialize, or rather to take on different forms and functions, essentially becoming different kinds of cells. And then finally there's morphogenesis, which is the sum total of all the cell differentiation, or rather the changes in shape and functions of the early embryo and fetus, and the resulting organs and tissues. Both differentiation and morphogenesis result in growth, thus increasing the number of cells. And this increase in the number of cells results in an increase in the size and mass of the zygote. This illustration shows the development of the morula and blastocyst. The morula is a solid mass of cells. Each cell is about the same size, and each has the capacity to develop into any of the many types of cells necessary to form a complete human. During this stage, individual or monozygotic twins can be formed. The blastocyst is composed of even smaller individual cells. As the cells continue to divide, they push outward, forming a hollow ball. At this stage, cells begin to specialize. So in the illustration, it's showing nothing but cleavages. So at day 1, the zygote is cleaved into 2 cells. At day 2, it's cleaved into 4 cells. At approximately day 4, we've entered the morula stage. At day 5, it's become a blastocyst. And finally, the last of these is simply the internal view of a blastocyst. Why they're showing it growing into what it already is, I'm not sure. The fertilized egg becomes a blastocyst. After the blastocyst forms, the cells begin to differentiate. Specific cells will form the placenta, certain protective layers, and the embryo itself. Plurally potent cells are at the center. These are stem cells which have the potential to become any type of adult cell. These cells and what are called totipotent cells, which simply means that these totipotent cells have an even higher capacity for differentiation. Both of these types of cells are the cells used in current stem cell research. The trophoblast, which is an outer layer of the blastocyst, surrounds the inner cell mass. This layer contacts the endometrial lining around day 7 and begins to release enzymes which digest their way into the endometrium so the blastocyst can implant there. The trophoblast releases the hormone human chorionic gonadotrophin, or HCG, which keeps the corpus luteum alive and is useful in detecting pregnancy. Or rather, if this hormone is present, it can be used to detect pregnancy. The blastocyst travels the length of the uterine tube and reaches the uterus in about 4 to 5 days. Once it drops from the uterine tube, the blastocyst floats freely in the uterine cavity for 1 to 2 days. At this point, the uterus is in the secretory phase, as endometrium is now producing nutritive fluids. The early embryo gets implanted in the uterus. A day after implantation, the blastocyst attaches even more firmly to the uterine endometrial wall. The cells of the trophoblast digest the endometrium, burrowing into the tissue. This attachment causes endometrial glands near the blastocyst to enlarge and increase their secretions. New blood vessels form to deliver even more blood to the implanted blastocyst, which it needs to survive. The trophoblast develops into the chorion. This is this fetal membrane will form the exchange membrane between fetal and maternal blood. The blastocyst placement, however, can sometimes cause problems. For instance, placenta previa. This is where the placenta grows near or over the cervical opening of the uterus, blocking the passage of the fetus during birth, and this can cause maternal hemorrhaging, which isn't good. The blastocyst inner mass then develops. During implantation and development, the blastocyst inner mass is still developing. Cells divide into two layers called the embryonic disc. The amniotic cavity forms between these layers. The yolk sac then develops in the intracellular mass around day 8. This illustration shows the primary events of the first week of embryonic development, or rather it's showing where the embryo is within the uterus and how much it's developed by the time it's gotten to that location, rather. So number 1, fertilization is the first phase, and this occurs within the uterine tube within 12 to 24 hours after ovulation. So number 2 is showing the cleavages. The first cleavage is completed around 30 hours after fertilization. The morula forms at around day 3 or 4. The blastocyst forms around 4.5 to 5 days after fertilization. And lastly, the implantation occurs about 6 days after fertilization occurs. Section 19.2, the embryonic stage is marked by differentiation and morphogenesis. So by day 14, the blastocyst inner cell mass has begun to split, which results in the formation of the embryonic disc, which contains two kinds of cells, the endoderm and the ectoderm. All of this occurs within the first 2 weeks before the unexpected menstrual period. It's estimated that upward of half of all conceptions actually do not result in successful implantation and pregnancy due to the hazards of the early embryonic stage. And a lot of this is simply because at this point the woman is not aware that she is pregnant. The embryo may not be receiving essential vitamins, minerals, or macronutrients, or may be exposed to toxins just because the potential mother is not aware of her pregnancy. Medical technology can assist in conceiving an embryo. In vitro fertilization, or IVF, mixes the egg and sperm in a dish. This method is used in 98% of all assisted conceptions. In vitro fertilization is also called pre-in vivo transfer. In this method, the ova are removed from the maternal ovaries and mixed with sperm, and then placed back in the mother's uterus after 2 days so that they can implant. This method results in twins 25% of the time and triplets 2 to 3% of the time, which is higher than what's seen in natural fertilization. ZIFT implants in the fallopian tube. ZIFT is zygote intrafallopian transfer. In this method, the zygote quickly implants into the woman's fallopian tube, the zygotes continue to mature until they reach the uterus, where they implant as usual. The last method is gift, in which the egg and sperm are mixed in the fallopian tube. So gift stands for gamete intrafallopian transfer. The ova are extracted from the woman's ovaries and the sperm from the man, and then inserted via catheter before conception into the woman's fallopian tubes. And in this method, any conception actually occurs inside the fallopian tube. Embryonic development takes roughly 6 weeks, and this is from week 3 through week 8. In this stage, the embryo undergoes rapid growth, differentiation, and morphogenesis. The rate of natural miscarriage in this stage is thought to be nearly 20%. Another thing which occurs in the embryonic stage of development is the development of a third cell type called the mesoderm, and this is the first landmark reached by the embryo. So now we have 3 layers, or 3 germ layers: the ectoderm, the endoderm, and the mesoderm. The ectoderm, as the name would suggest, is the outer surface of the embryo. The epidermis, the entire nervous system, portions of the eyes and teeth, the posterior pituitary gland, and the adrenal medulla develop from this layer. The endoderm is the innermost layer. This produces the alveoli, the liver, most endocrine glands, the tonsils, portions of the inner ear, and the epithelial linings of the digestive tract. And then finally there's the mesoderm, which is the layer between the endoderm and the ectoderm. The dermis, all of the connective tissues, including the skeletal system, muscles, blood, kidneys, testes or ovaries, the reproductive ducts, and the lymphatic vessels, develop from this layer. And it's probably a good idea to really pay attention to what develops from each of these layers, because they really don't follow much of a theme. This illustration shows embryonic development from day 14 to around day 35, and here's just labeled all the parts of the embryo at certain stages of development. There's the amniotic cavity, the embryonic disc, the yolk sac, the blastocyst cavity, the trophoblast, the chronic villi, the allantosis, the body stalk. Eventually, we form the amnion, and also it's begun to form the digestive tract at day 35. At day 35 also, it's clearly got the ectoderm, the mesoderm, and the endoderm labeled, and also what's developed into the umbilical cord. And we'll cover all of these in greater detail throughout the rest of this chapter, and hopefully by then we'll have cleared a lot of this up. The extraembryonic membranes develop into essential carriers of nutrients. So now we're going to begin going through all of those parts of the embryo that I just showed you. The amnion lines the amniotic cavity. The amnion provides a diffusion area for amniotic fluid, which is a fluid derived from the maternal interstitial fluid, cleansed of embryonic and fetal waste products, by diffusion across the amniotic membrane, or rather the amnion is cleansed. The amnion protects the developing fetus from injury, allows free fetal movements, maintains a constant temperature, and permits lung development, and helps to remove fetal urine. The allantosis is actually the start of the umbilical cord. It provides the starting material for the blood vessels of the umbilical cord. These vessels will transport fetal blood to and from the placental exchange surfaces where oxygen, nutrients, and waste materials are diffused across this membrane. The yolk sac will eventually become part of the digestive and respiratory tracts. This yolk sac produces fetal blood cells up until the point the bone marrow forms, and may also be involved in gamete production. The chorion is the source of HCG, or human chorion gonadotrophin. The chorion is the outermost layer of the extra embryonic membrane and develops from the trophoblast to make up the exchange portions of the placenta. Human chorion gonadotrophin, or HCG, like I said earlier, is the hormone that maintains pregnancy until the placenta is fully functional and helps to prevent against the degradation of the corpus luteum, which we'll remember is a temporary gland within the ovary which helps to maintain pregnancy by releasing hormones. The placenta is essential but disposable. The placenta is necessary for embryonic and later fetal development, essentially because it acts as a diffusion surface for oxygen, nutrients, and waste products. Within the placenta are these structures called embryonic chorionic villi, which extend into the endometrium of the uterus, and structurally these are similar to the villi seen in the small intestine, as one would expect. Both these villi and the endometrium are the precise structures within the placenta that act as diffusion surfaces for oxygen, nutrients, and waste products, and help to ensure that fetal and maternal blood don't mix. The placenta also produces hormones which help to maintain pregnancy. So early in pregnancy, the placenta secretes human chorionic gonadotrophin, which stimulates the corpus luteum to remain viable. Progesterone and estrogen from the corpus luteum are what prevent the loss of the endometrium, so that implantation of the embryo can occur successfully. So over here in this illustration at right, you can see a closer view of the placenta, and it's divided into the maternal portion and the fetal portion. And in the fetal portion of the placenta is where the chorionic villi reside, and then the maternal portion of the placenta includes the endometrium. And I'm not really going to go over this much, but you can see that the placenta acts as this diffusion surface for the maternal, for the oxygen, nutrients, and waste products, and ensures that the maternal and fetal blood don't mix. And it also eventually runs into the umbilical cord, which contains these umbilical arteries and veins to ensure that, or rather the job of these arteries is to deliver the oxygen and nutrients to the developing embryo or fetus, and then to remove the waste products, which then diffuse in the other direction of the oxygen and nutrients so that they can be disposed of by the mother. Gender differentiation occurs at approximately 7 weeks. So prior to week 7 of development, male and female development is exactly the same, with 2 distinct sets of reproductive tubes and no differentiation between the sexes. However, by week 7, sexual differentiation begins to occur, and this is essentially dependent upon if a Y chromosome is present or not. So if a Y chromosome is present, the male tubes will be stimulated, the testes will develop, and release of testosterone from the new testes will then cause male sexual characteristics to form. However, if no Y chromosome is present, the organs destined to develop into the ovaries will mature instead. So all of this is shown in the illustration at right. So up at the top is the undifferentiated stage. In the illustration, it's about week 5 of embryonic development. And at this point, the genitals are called the genital tubercle, and the various parts are labeled on this drawing of this undifferentiated sexual, or rather genital organ. However, by week 10 of embryonic development, we begin to see the differentiation into the male and female genitalia. And at this point, there isn't a whole lot of difference between the 2 sets of genitalia. However, near birth, we have complete differentiation of these genital organs into the very recognizable genitals associated with males and females. The embryo becomes increasingly human. At the end of the embryonic period, the newly forming individual is approximately 2.5 centimeters or 1 inch long and has a recognizably human form. All of the internal and external structures are present at the end of this phase, and the placenta is fully functioning. And this is all depicted in the diagram at right. However, I'd like to draw attention to the key in the middle of this diagram, because a lot of these measurements are completely inaccurate. For instance, if you look at 32 day equals 1 centimeter or 2.5 inches, the inches and centimeters are actually reversed. That should be 2.5 inches, or rather 2.5 centimeters equal 1 inch. And I think this mistake is found throughout the key, because 2 centimeters is not equal to 5 inches, because 5 centimeters is equal to 2 inches. So they just have the centimeters and the inches backwards. And I'm probably not going to really test you over this diagram much because of that mistake. But anyway, you can see at day 20 of the embryo's development, it's pointing out the neural plate and the neural groove, and also some of the other parts. Essentially, what's forming here is the beginning of the spinal cord. So by day 28, we actually have the beginnings of the spinal cord, essentially. And then by day 32, we begin to see you can obviously tell that it's going to be a living organism by this time, though I kind of disagree with the book in that you can tell it's going to be a human at this point, because it really doesn't look like one to me. And then by day 52, you can definitely tell that this developing fetus is definitely human, and they've labeled some of the more obvious parts here in this illustration. Section 19.3, fetal development is a stage of rapid organ growth and also maturation. The fetus begins this stage at approximately 2.5 centimeters long and weighs approximately 1 gram. Within 7 weeks, the fetus will grow to an average length of 50 centimeters or 20 inches and weigh about 3.75 kilograms or 8 pounds, which I believe is a little bit higher than average for the weight. The mother's body will begin to show signs of the expanding uterus during the fetal stage, and over in the illustration at your right, you can see just how much space the womb takes up during pregnancy, so much that it's actually pushing up against the small intestines and down on the bladder. And there's really not much else there's really not much else I want you to take away from this illustration other than the placement of the womb during pregnancy and how much it expands. Fetal circulation is unique. Nutrients from the mother enter the fetal circulation across the placenta by diffusing down their concentration gradient, just as fetal wastes leave by diffusing down theirs. So here we have just simple diffusion again, as substances always diffuse down their concentration gradients. Oxygen carried by the mother's hemoglobin is taken from the maternal hemoglobin by fetal hemoglobin, which has a higher affinity for oxygen. Or rather, fetal hemoglobin more readily takes up oxygen gas than maternal hemoglobin. Fetal urine is then the main source for replenishing the amniotic fluid. The volume of amniotic fluid rises throughout pregnancy and peaks around week 33 at about 750 milliliters. This volume is regulated by absorption into the maternal bloodstream and by fetal respirations, which is where the fetus swallows small amounts of amniotic fluid continually, which sounds kind of gross seeing as there's a bunch of urine in it. So here's how placental and fetal circulation works. The fetal blood leaves the heart and moves through the umbilical arteries to the chorionic villi, where the associated gases and waste products diffuse with the maternal blood. The blood is collected in placental veins and returned to the fetus through the single umbilical vein. The umbilical vein then travels to the fetal liver, where it is dropped into the hepatic valve. The round ligament is what marks the path of the umbilical vein from the navel to the liver. The umbilical arteries dissolve after birth and add to the hepatic capillary system, and you can see the flow of blood from the placenta to the umbilical cord and to the developing fetus in the illustration at right, which I'm not really going to go over much. Just more or less know the flow of blood from the placenta to the fetus and then back again. Fetal development is the longest stage of pregnancy and occupies both the second and third trimesters. Pregnancy is typically divided into trimesters, which essentially divides the length of pregnancy into 3 distinct parts. The first trimester includes all embryonic development and the first month of fetal development. The second trimester has 3 different parts, essentially. In month 4, we see continued rapid changes. The face begins to resemble its final form at birth. Blood cells are produced by the liver and the bone marrow. By month 5, the nervous and muscular systems have developed enough that by the 5th month, movements may begin. Quickening occurs, and this is referring to the fact that the fetus has begun to move. The fetal skin is covered in soft hair called lanugo. At the end of the 6th month, the fetus weighs approximately 450 grams. The lungs begin secreting surfactant, and the fetus could potentially survive outside the womb at this stage. The 3rd trimester is characterized by continued rapid growth and maturation. The eyes open, the suckling response begins to develop, and loud noises can initiate a startle reaction. The fetus often moves regularly. A layer of protective fat begins to develop. This is called brown fat, and in males, the testes descend. At the end of week 38, the fetus is prepared for life outside the uterus, and it should have maneuvered to a head downward position, and the fetus should have dropped at this point. This illustration shows some stages in fetal development, and I'm not really going to go over this much either because I feel that it's pretty much self-explanatory. But you can see at week 4, the human fetus looks like this non-human thing, which may or may not even develop into a human if we're just to look at it. And then by week 36, we have what looks like a normal human baby, even though this picture is kind of weird looking, so it's still an abnormal looking baby, I think. So here's a chart which summarizes fetal development, and it's been divided into the embryonic period and the fetal period. And if you want to go over this, I recommend that you just pause the video and go over it yourself, because there's just way too much information here for me to go and read it. But this should be a helpful study aid, and I'm going to try to put at least 1 or 2 questions over this chart on the exam. Prenatal analysis helps parents and medical professionals. Obstetric visits typically occur at 18 to 20 weeks in the United States, and these routinely include an ultrasound examination. The gestational age can be determined by measuring and comparing the size of various fetal body parts. The heart can be seen beating. Internal organs and the skeleton can also be viewed, and at this point usually gender can be determined, though sometimes gender is determined wrong at this point. In amniocentesis, this is the collection of the amniotic fluid for analysis. This is routinely performed at 15 to 18 weeks in order to determine gender without a doubt, and also to look at the condition of the chromosomes. The DNA within shed cells, is isolated in a laboratory, and a karyotype is created. A karyotype is just a visual representation of all 23 pairs of chromosomes, with the homologous chromosomes being paired together and ordered from 1 to 22, and then they'll have the sex chromosomes off by themselves. And with karyotypes, you can sometimes tell if there's a genetic disorder that the fetus has, such as if they have Down syndrome, they'll have 3 copies of chromosome 21 when they should only have 2. Or you can tell if there's some sex-related disorders such as Turner syndrome because they'll only have 1 X chromosome, or Klinefelter's syndrome because you'll have 3 X's and then a Y. Or maybe, or rather I should say 2 X's and a Y is usually what occurs, sorry. The chorionic villus sampling is used to detect genetic anomalies earlier in the pregnancy. And then there's finally 4D ultrasound, which is a computer-enhanced ultrasound which produces a clear, lifelike view of the uterus. And in the illustration at your right, this is showing an amniocentesis, where they'll take a needle and stick it into the womb to extract the amniotic fluid for analysis. Section 19.4, labor initiates the end of pregnancy. Labor and birth mark the end of prenatal life and of total reliance on the mother. The fetus goes from an aqueous environment with total life support to a dry environment where all life functions must come from within the infant itself. All organ systems must be in place and be fully functional at this point for life to continue. Labor begins with hormonal triggers. It's also one of the few positive feedback loops seen in living things, so that's unique. Labor begins with hormonal triggers that are thought to originate in the pituitary gland of the fetus. So the fetal anterior pituitary gland secretes ACTH, which triggers the fetal adrenal gland to secrete hormones that affect the placenta. The placenta then increases production of estrogen and decreases production of progesterone. Estrogen in turn increases oxytocin receptors on the placenta and increases placental prostaglandin production. This combination of factors makes the uterus much more sensitive to oxytocin levels. The maternal oxytocin then initiates rhythmic contractions in the uterus. Like I said, this is an example of positive feedback, as contractions of the uterus stimulate oxytocin production. More oxytocin means more and harder contractions, and more oxytocin, or rather even more oxytocin I should say, the contractions become stronger, harder, and closer together. Most births actually take 24 hours from initial contraction to delivery. So over at your right, you can see how this positive feedback system loop work. Contractions of the wall of the uterus force the baby's head or body into the cervix, thus increasing the stretching of the cervix, which causes the stretch-sensitive nerve cells in the cervix to send an impulse to the brain. The brain then interprets the input and releases oxytocin, which in turn causes the muscles in the cervical wall, or rather the uterus wall, to contract more forcefully. The baby's body stretches the cervix even more. And as this is a positive feedback system, the increased stretching of the cervix causes release of more oxytocin, which results in even more stretching of the cervix. Interruption of the cycle causes the birth of the baby to decrease stretching of the cervix, and this will, of course, eventually break the positive feedback loop. Delivery has 3 stages: dilation, expulsion, and the placental stage. In dilation, the baby's head presses on the cervix. The pressure and the uterine contractions stretch the cervical opening from slightly less than 2 centimeters to over 10 centimeters. The mucus plug falls out of the cervix, and yes, there is a mucus plug in the cervix during pregnancy, which ensures that the amniotic fluid stays where it needs to be. However, when it falls out, the amnion membrane ruptures, releasing the amniotic fluid, and this is what occurs when the water has broken or the water breaks. During expulsion, this is the time from the full cervical dilation to delivery. So in this stage, the uterine contractions gain even more strength, and the mother assists in the birth by pushing voluntarily with muscles. The baby then moves through the cervix and out the vagina. And finally, in the placental stage, the placenta and umbilical cord are also expelled through the birth canal. The arrangement of the placental vessels may suggest the presence of congenital birth defects. So over here in the illustration at your right, you can see these 3 stages: dilation, expulsion, and the placental stage. And to me, the expulsion stage looks kind of painful because the baby's neck is kind of twisted in an unnatural position, but I'm not sure how accurate this illustration is, and I won't go over these again because all this information is in the slide. Fetal development can have many complications. The baby may be premature if birth occurs before 37 weeks of gestation. These premature babies require specialized care in neonatal intensive care units until their organs have fully matured. Many medical professionals believe that industrial chemicals, pesticides, poor standards of living, and air pollutants lead to premature births. Labor may need to be induced or forced, and this is what needs to occur when the mother has went over the prescribed amount of gestation to move the baby out of the uterus. The baby may be breached or unusually positioned in the womb, and usually this involves the baby's not being turned into the right position where the head is pointed downward. However, babies or fetuses in the breached position can be turned using gentle pressure from outside the uterus. A cesarean delivery may be needed, and this is where they would cut open the mother's abdomen and womb and just remove the baby directly out of it in cases where the baby can't be forced out the birth canal or if some other problem is occurring. Some infants fail to gain weight after birth for whatever reason, which can be quite concerning to parents, I would imagine. And sometimes there is ambiguous sex determination as the genitals haven't developed to a point where the baby is definitively male or female. After the infant has been born, it will need to be fed, and this is why the mammary glands provide milk when needed. So no milk is produced by the mammary glands until after birth. The hormone prolactin is first secreted by the anterior pituitary gland, and this hormone is what causes the mammary glands to secrete milk. Oxytocin is the hormone released due to infant suckling, and yes, this is the same hormone which was causing a cervical contraction. It actually stimulates the let down response, which causes the contractions of the larger lactiferous ducts. Colostrum is essentially a special kind of milk produced 2 or 3 days after delivery. It's actually a very watery fluid that is rich in proteins and antibodies, which will help to increase the baby's immune system abilities. There are several benefits of breastfeeding. The antibodies and lymphocytes present in breast milk help to protect infants from numerous diseases during the first months of life, as they likely would have a reduced immune system due to the fact that they haven't been exposed to many pathogens. And over here in the illustration at right, you can see how the hypothalamus directs the anterior pituitary gland to release prolactin and oxytocin to ensure the secretion of milk, and then the suckling of the infant actually signals the hypothalamus to begin the cycle all over again. And this is the last slide in this module, and it should be, well, I'll just say this right now, my next slides likely do not have recorded audio over them, just because I don't think anyone's really going to take the time to listen to them. So this is actually the last set of slides in this class. So just study and do well on this last test before the final, and also study for the final course.